Omega-3 fatty acids, particularly eicosapentaenoic acid, EPA, and docosahexaenoic acid, DHA, are essential nutrients with well-documented benefits for cardiovascular health, inflammation control, and cognitive function. However, emerging research is revealing an even deeper role for these fats in maintaining brain health through the modulation of autophagy, a cellular housekeeping process critical for neuronal survival, resilience, and long-term function. Among the various tissues in the human body, the brain is perhaps the most vulnerable to the accumulation of damaged proteins and dysfunctional mitochondria due to its high metabolic demands and limited regenerative capacity. It is here that omega-3 fatty acids demonstrate some of their most promising effects by promoting autophagy-linked neuroprotection and potentially helping to prevent or slow neurodegenerative disorders such as Alzheimer's disease. At the molecular level, EPA and DHA influence several key signaling pathways involved in autophagy, including the inhibition of the mTOR pathway and the activation of AMP-activated protein kinase, AMPK, and transcription factor EB, TFEB, a master regulator of lysosomal biogenesis. These changes promote the initiation and progression of autophagy in neurons, allowing the brain to clear intracellular waste products, including misfolded proteins like beta-amyloid and tau, both of which are strongly implicated in the pathology of Alzheimer's disease. By enhancing autophagic flux, the complete cycle from autophagosome formation to lysosomal degradation, omega-3s help maintain the cellular environment necessary for neuronal communication, plasticity, and repair. DHA, the most abundant omega-3 in the brain, is particularly effective at integrating into neuronal membranes, where it contributes to membrane fluidity, signal transduction, and synaptic integrity. Research shows that DHA not only promotes autophagy, but also activates neuroprotective gene networks that reduce inflammation and oxidative stress, both of which can impair autophagic function if left unchecked. Through these complementary mechanisms, DHA helps maintain a favorable intracellular environment for ongoing cleanup and regeneration. EPA, though less concentrated in brain tissue, has its own unique anti-inflammatory properties and supports systemic metabolic conditions that indirectly enhance autophagy in the central nervous system. One of the most compelling aspects of omega-3 induced autophagy is its potential to act preventively before neurodegeneration takes hold. In animal models of Alzheimer's disease, supplementation with DHA has been shown to reduce amyloid plaque deposition, restore synaptic markers, and improve cognitive performance. These changes are closely associated with increased expression of autophagy-related genes and better lysosomal function. Moreover, some human studies suggest that higher omega-3 intake correlates with slower brain aging, improved memory retention, and reduced risk of Alzheimer's and other forms of cognitive decline, outcomes that may, at least in part, be mediated by improved autophagic efficiency. From a nutritional standpoint, obtaining therapeutic levels of EPA and DHA is both achievable and sustainable through dietary sources and supplementation. Fatty fish such as salmon, sardines, mackerel, and anchovies are rich in these long-chain omega-3s and offer additional benefits such as high-quality protein, selenium, and vitamin D, all of which contribute to neurological health. For those who consume little or no fish, purified fish oil supplements or vegan algal oil capsules can provide a reliable and concentrated source of DHA and EPA. Consistent intake over time is key, as omega-3s must be integrated into cell membranes and accumulate in tissues to exert their full protective effects. Importantly, omega-3 fatty acids also work synergistically with other autophagy-promoting practices and nutrients. When combined with regular exercise, intermittent fasting, or polyphenol like curcumin or resveratrol, EPA and DHA may enhance the cellular response to metabolic stress and deepen the neuroprotective effects of autophagy. In this way, omega-3s function as part of a larger toolkit for maintaining brain resilience, addressing not only the structural needs of neurons, but also their dynamic capacity for self-repair. What sets omega-3s apart in the context of neuroprotection is their ability to act at the intersection of metabolic regulation, inflammation control, membrane integrity, and autophagy. Each of these domains plays a vital role in the prevention of age-related cognitive decline, and the integration of omega-3s into the daily diet represents a foundational step toward protecting the brain against the cellular damage that accumulates with age. This is not just about delaying disease onset, but about actively supporting the brain's inherent capacity for renewal and adaptation across the lifespan. In conclusion, omega-3 fatty acids, particularly DHA and EPA, provide powerful support for autophagy in neurons, offering a practical and well-supported strategy for enhancing brain health and preventing neurodegenerative decline. Through mechanisms involving YMPK activation, mTOR suppression, and improved lysosomal function, these essential fats help clear cellular debris, reduce inflammation, and support long-term cognitive resilience. Whether consumed through fatty fish, high-quality supplements, or algae-based alternatives, omega-3s stand out as one of the most accessible and scientifically grounded ways to support autophagy and protect the brain in the modern age.